Good morning, good night, and good day, depends where you are. And pretty sure audience from everywhere, including our panel speakers today. It's my great pleasure today to introduce Bioverse Episode 9, the essential role of CMC in the global drug development and the deal making of ADC, radiopharmaceutical, and cell and gene therapy. No need to say, all those stuff are super interesting to the current global drug development industry. That's why we have three renowned experts in this area to share their insights and intelligence and visions with all of us. Before we get started, for those who just joined Bioverse the first time, let me share with you who we are and what's our mission. Here we go. This is today's title. Like I mentioned, I don't need to repeat. And this is a two major organizer of Bioverse webinar. It is a co-production by SAPA-GP and Incense We Trust community. SAPA-GP is a Philadelphia chapter of a Sino-America Pharmaceutical Professionals Association with thousands of members, million followers, multiple chapters, and a lot of footprints. So for SAPA-GP, one of their signature conference is cell and gene therapy, which is really relevant to what we'll talk about today. Regarding Incense We Trust community, we are also a 501c3 nonprofit based in New Jersey. Have 2,000 members, and half of them come from North America, 40% from East Asia, and the rest is from all the major biotech hub in the world. And we are proud to have a huge number of C-level executives from Asian biotech communities. We do publish a weekly newsletter, and of course, we do the monthly webinar. That's what we are doing today. Now, the question is like, why we are doing the CMC in ADC, radiopharmaceutical, and the CGT today? I want to remind people who are not living, breathing in this world, give you a little bit idea why this is such a challenging topic and also could be a competitive advantage. So at least three highly visible news in the past 2 or 3 years. The first one, we all know, one of the blockbuster ADC was a Trodelvy, came from Immunomedics, but it was not easy. So that approval was delayed for more than 1 year because of the problem they have with their manufacturing in their New Jersey site, not far away from where I stand, actually. That's the first one. That's ADC. Second one, we all know CAR-T right now is on fire. And Abecma just received this approval for the third line multiple myeloma. But, if you look back, Abecma had quite a bit hiccup because of the CMC, and they actually got a refusal to file later from the FDA. And that delayed the submission of this CAR-T for multiple myeloma for almost a year. The last one very recently, Pluvicto, right now is on the trajectory to become a blockbuster drug. It's kind of probably going to in the near term best-selling radiopharmaceuticals, had the same issue. As you can see here, their manufacturing become bottleneck for them to really ramp up their commercialization, even after they had received the approval from the FDA. With that being said, now I hope everyone understands why CMC, by the way, I should give the full name, which means chemistry, manufacturing, and control, is so important for the drug development, especially for those complicated modalities such as ADC, radiopharmaceutical, and cell and gene therapy. With that being said, it's my great honor to let the three experts to introduce themselves. If you don't mind, could you please introduce yourself from the left to right? So Dr. Fan, do you want to go first?
Sure. Hello, everyone. My name is Jing Fan. So I'm the founder and the CEO of Hopstem Biotechnology. So we are a startup since 2017, founded both in Baltimore, US, and also Hangzhou in China. So we're a company focusing on developing iPSC-derived cell therapy for CNS diseases. Personally, I graduated from University of British Columbia in Canada with my PhD in neuroscience. And then I did my postdoc in Institute of Cell Engineering at Johns Hopkins. That's where I started my work using pluripotent stem cells to derive different human neuron cells for drug screening, as well as transplantation for stroke models. And that's when, about 2016, I see the field is becoming the future as cell therapy for CNSs, which a lot of those indications were large unmet medical needs. So I decided to start up a company and trying to transform those technologies and for those patients. And thank you, everyone. Yeah, hope we can enjoy a conversation useful for everybody here. Yeah.
Fantastic. So Jimmy?
Sure. Thank you, Leon, and good day to everyone online. My name is Jimmy Lee, CEO of WuXi XDC. WuXi XDC is a CRDMO dedicated to serve ADC and bioconjugate industry. So we provide complete coverage for ADCs and bioconjugates. Personally, I'm a biochemical engineer by training with a bachelor's from Tsinghua University and PhD from University of Maryland, Baltimore County. Worked in the US, San Diego for 10 years. I was with Genentech before joining WuXi Biologics in 2011. In about 13 years with WuXi, I started basically setting up the cell culture process development lab in WuXi and managed the team for about 5 years. And I moved on to manage manufacturing. I was actually responsible for WuXi Biologics' first BLA approval, as well as the first EMA approval. And that manufacturing for about 4.5 years. And then in late 2020, we started to integrate the capabilities to serve the ADC industry, which led to the establishment of WuXi XDC in 2021. And XDC also gone public actually last year in November 2023. So we are now also a publicly traded company in the Hong Kong exchange. Looking forward to interactive and great discussion with the folks online. Thank you.
Last year, Gary.
Hi, hello everyone. My name is Gary Lee. Really glad to have this opportunity to serve as a panelist for this meeting. So I have about 20 years industry experience in both large pharmas and small biotechs. First 10 years working for Pfizer and Johnson & Johnson, mostly in oncology, drug discovery, and early development. Recent 10 years, I've been involved with 3 successful startups. First, Ignita, IPO, then acquired by Roche in 2017, and then BridgeBio, and most recently, RayzeBio. RayzeBio joined as a 3rd employee and senior VP, led the discovery, translational medicine, and portfolio strategy from its very beginning. And after recent acquisition of RayzeBio by Bristol Myers Squibb earlier this year, I have been running my own consulting firm, advising startups and venture capital firms. And you might notice that last week I joined on a part-time basis a company called AdvanceCell as a chief business officer. So again, really great to be here to talk to discuss. So apparently I'm not a CMC person. Maybe I'm the only one that's not really CMC. But I'd like to provide my own perspective and opinion on this topic if it's appropriate. So thank you very much.
Right, fantastic. Now I guess everyone in the audience and those around the world understand why we invited 3 experts that definitely have a lot of decades of experiences in their individual field. So with that being said, let's dive into this. Today, actually, we're gonna have 3 major buckets. I want to give people a little bit kind of idea what we're gonna talk about. First, we're gonna talk about how CMC in this 3 modality play such an important role in the global drug development. That's number one. Number two, we're going to talk about how CMC play a role in the deal making on the global stage. And last, we would invite the three speakers to share some, we must know, but often overlooked aspect of each of those modalities. With that being said, let's dive right in. The first one, the essential role of CMC in global drug development, including both drug discovery, preclinical, and clinical, and submission and commercialization. Probably let's start with the first one. I would like to ask Gary, we chat this a little bit. What's a unique constraint for a complicated modality like radiopharmaceutical for during drug discovery phase? If you could share, a little bit idea what's special about radiopharmaceutical, what's interesting aspect you have say the challenge, that would be fantastic.
Sure, I'd like to kick this off. Before I address the pivotal question that Leon raised earlier, I'd like to first just briefly talk about radiopharmaceutical in general. So just give you the context so we can see why CMC and it's so critical for radiopharmaceutical. So there are basically two different types of radio isotopes used for imaging and therapeutics. But there's a so-called radiohalogens, for example, the iodine, the astatine, etc. So those radioisotopes can be linked directly to the binder. And the other type of radioisotopes called radiometals, such as lutetium, actinium, lead, copper. The more popular type of radioisotope used recently. So those radioisotopes have to be chelated and then linked to the binder through a linker. So in a way, similar to the ADC, right? Think about you have a binder, linker, and payload. Here we have a binder, we have a linker, we have a chelator, which will chelate the radioisotope. So the binder can be antibodies, can be nanobodies, can be antibody fragments, can be peptides, or even small molecules. The challenge here, first of all, it's a very complicated drug. Secondly, you have the radioisotope, which decays. So the issue with the radioisotope decay is that you cannot make your final drug product beforehand. There's really no real shelf life to speak of. For example, even for the long half-life radioisotopes, such as lutetium, actinium, they have about 8 to 10 days decay half-life. So basically you have 3 to 4 days, maybe maximum 5 days between the time you make your drug to the time that the drug has to be given to the patients. So, and on top of that, once you have your drug product made, there's a phenomenon called radiolysis. Basically, your isotope start to destroy your drugs. So you can't really put this on shelf. It's made on demand in a way. Because of this, so it's very critical to have your drug, as we call precursors. Basically, it's a binder, linker, and empty chelator ready as a precursor, and you have your radioisotope precursor, and you put them together at the time at prescription, and you put the drug together. You do your quality control, and you ship to the patients, and then give the patient within a few days. To do this for the longer half-life isotopes, probably you can use FedEx or any global carrier to ship. For the shorter half-life isotopes such as lead or astatine, you have 10-hour half-life. So basically from the time of the drug to patient, you have maybe 5 hours or maximum 8 hours, depending, the stability and depending the injected activity. So there's a big challenge in terms of how to really make the final product, how to ship the product, and how to deliver this to patients. So going back to the question that Pluvicto had back then, part of the reason was that they only, back then, Novartis only had one manufacturer, commercial manufacturer that can produce a drug, which is in Italy. You can imagine that any disruptions in the supply, transportation, natural disaster, weather delay, anything, or radioisotope supply, any of those can affect the manufacture. And also because the drug has to be given in multiple cycles. And once a patient are already on the drug, you need to prioritize that patient for second, third, fourth dose. So they have to stop the new patients because of that. Solution, right? So as we know, every drug discovery or development, we need a redundancy. So that's important because things will go wrong, as you know. So the relatively easier solution, just to have more sites, more manufacturer sites. Certainly, it's easier said than done because for the radiopharmaceutical, the regulatory requirements, the supply, the cost, everything's it's more challenging than a typical small molecule-based drug. Luckily, I think Novartis was, as a big pharma, able to do that. They established a second site, and I think maybe third site in Europe sometime soon. Hopefully, with additional sites, with more steady supply of lutetium. So they are right now. I think they don't have the problem anymore. So. But we can dive into more detail later because I think there's some questions coming up as well. So.
Yeah, fantastic. I'll throw some suspense right there because as you probably recall, Novartis just acquired another company last week. And so, we'll get into this in our second bullet points about the deal making. But with that being said, any question from the panelists, like Jimmy and Jean, any interesting you want to learn before we move on to the next one?
No, it's great to hear Gary speaking about the challenges of radio isotope related drugs. It is actually also part of what we call the XDC field, RDC, and so it has some similarities to ADC in terms of the challenge. In terms of ADCs, obviously a lot of the payloads are highly potent, highly toxic. So the handling there, not because of the short shelf life, but the handling itself, requires a lot of safety precautions. And the CMC part, it's also very complex because it's chemistry meets biology. So that's first time in history when you make a drug by combining two different modalities together. So there are a lot of complexities there too.
Fantastic. So probably now let's just learn a little bit more about the CGT. So Jean, congratulations. First of all, I heard that your IND was just accepted by the FDA. And if you could walk us through, the case you had in your hand and what's the general rules you can, share with the people who are especially interested in CGT field, especially CGT clinical trial IND submission in the US, regulated by the FDA, that would be fantastic.
Yeah, thank you, Leon. Yeah, so actually from our case, the iPSC allogeneic cell therapy, we would say it's a long journey. I think we had the interactive meeting with FDA since 2019. And at that time, there's already some companies in the field developing iPSC cell therapy. So we're lucky when we talk with FDA, we got some clear guidance that you cannot be found in the current guidance out there. It's only when you talk with FDA and you have something already developed, they can talk to you. If you haven't done any work, there's no way you can get those guidance. So we start early, and we have to say, there's always a saying, say, for cell and gene therapy, most of the work, say 80% is about CMC. That's how important it is compared to, other biologics. And 20% is clinic. For the other drugs, they think it's the other way around. I wouldn't say exactly I agree with this because it seems like the clinical work for cell therapy is less challenging that way. But actually, I would say for clinical work, we are probably comparable or even more challenging in determining the cell therapy potencies in clinic, right? And for the final BLA. So even if we say it's equal with antibodies and other biologics, for clinical work, we could say the CMC for cell therapy could be 3 to 4 times workload than the normal ones. Because for traditional drugs, there's has been a long history and a lot of analytic method and processes has been settled. So it's only the innovations you're dealing with. But for us, it's so innovative. There's no certain path we can follow, including those as CAR-Ts. It's quite different. So at that time, we have to start from early as donor screen, especially we're doing products that we're aiming for global market. So first we do, both in AMPA and also FDA application, and we have to consider the differences between regulations, even including EMA. And that time there's no clear guidance or, no industry standards you can follow. So we have to develop our own, and we do join the conversations and trying to contribute of what we know about those products to the guidances and regulations. Yeah, so let's say when we submit our IND package, that's where after the pre-IND meeting by the end of 2022, although we have to work with a CDMO partner in US to make sure they can produce the same drug product by formulation, same as we did in China. Because in China, we have a full GMP facility, and the team, we have worked on these CMC for 4 to 5 years. And it's a, quite a long way, and especially consider a product, the differentiation manufacturing itself takes about 42 days. And we're not a super long iPSC cell therapy product. There's, other product that could take 6 to 7 months for the manufacturing itself. So I can't imagine how those products will eventually, going for commercial manufacturing. It's quite challenging, especially thinking about cell products. They are live drugs, right? So it's quite interesting. Like first, you have to be careful with all the viral and microbial contaminations from the beginning, even from the donor screen. And it could be from the soil of those tissues, and also can be introduced from the process of manufacturing. And so that's why for the GMP facility, we're emphasizing so much on how you, even doing the early development, you have to make sure your cell bank and everything qualifies, and eventually for the BLA. You don't want to do too much work on those cell banks, and with your product, even on clinical trials, where later on you find out those cell banks won't be qualified for BLA. And also, cells are also fragile. Like Gary said, before we always think cells is hard to do the logistics because their shipping, storage, and everything needs extra care. First, we don't want to have any contamination during the shipping, and you have to have a closed system. And we're developing closed system manufacturing. Eventually that's better for the commercializing. But also for shipment and storage and how stable they are, you have to test out. So the stability study is covering the whole development. And also, because they're fragile, you don't know after those shipping storage whether they are still the potent product as you expect. So it's not only about the safety, it's about the potent as well, especially when we received the clearance with FDA. We found it has been a lot of work we have done. So there's not too much question about CMC, but they're really emphasizing on how you're going to deliver those cells. Say we are treating chronic stroke, so we have to do an intracranial injection, and we have to make sure all the equipments and other stuff we use along the administration of this drug, they won't bring extra, safety issue, and we have to prove, give the evidences. And since a lot of question has been answered during the pre-IND meeting, so we receive really less questions during IND, but we had a huge package. I think the seniors, our third party, for registration, just mentioned to us during their publication for the ECTD of the package, they realized how huge the work it is. It's, super long IND files. It's only, it's like for normal INDs, it's maybe several hundred pages, and for us, it's several thousand pages. And even for one attachment for M3, it's 200 pages. that single, for each single one attachment. And we have more than 200 attachments. So we can only say it's too much work with iPSC. Even comparing to CAR T, people think it's combination of gene therapy and cell therapy. You have to provide a lot of CMC materials for the virus itself, right? For CAR Ts, but for iPSC, we just have to say we probably double or triple CAR T's work. Yeah, but it's worth it. I think as long as we see, the efficacy primarily in the patients we have in China trial. So we now think, it's a stable CMC for now, for current stage, but eventually we have to go and improve and prepare for the commercialization. And that's where we learn from other biological drugs and probably as scary for radiopharmaceutical drugs as well. Yeah. So yeah, thanks. That's how much I would like to share for now. Yeah, thank you. Mm-hmm.
Oh, that's a lot of very interesting insights and, something I don't know before today. And also, I think like what Gary and Jean shared is a natural transition to actually my question. Obviously, it sounds like this is really complicated and challenging, a lot of investment, capital, technology. Obviously, that probably going to scare away a lot of biotech. But don't worry, I think there's someone can come to help, which is going to be WuXi, right? WuXi, your CRDMO. I think you're really help people. If you have innovation, you kind of, mitigate their risk, reduce their burden in terms of, the research, drug discovery, development, even the clinical development, and also the later submission in terms of a CMC. So from that perspective, Jimmy, can you share some of your view how you are enabling the small biotech particularly to really get into those spaces? Of course, you are focused on ADC, but if you can talk broadly, how you help them, through the drug development and also submission for the marketing approval, that'd be great. Thank you.
Thank you, Leon. I think it's a great question, right? I think a lot of the audience online today are probably biotech scientists or executives. A lot of the biotech executives are scientists by background, and obviously drug development is very different than drug discovery. It's a marathon process, right? That takes a lot of expertise. So it's actually very often that a scientist that discovers or has great idea on the drug, but probably has very little knowledge or understanding or expertise on how to commercialize the drug in terms of making it stable, high quality, and affordable, right? So that's what we do basically as a CRDMO is we help turn scientists or entrepreneurs' ideas into a consistent, robust, scalable process. So in terms of ADC, it starts actually at the discovery stage, because as I just mentioned briefly, ADC is where chemistry meets biology. So even at the discovery stage, a lot of our clients actually will need support because they might be an antibody-based company who does not have much knowledge on payload linker, right, which is a small molecule, or a chemistry-based company who has a lot of expertise on payload linker but doesn't have much experience on antibodies. So for ADC, even at discovery stage, we found that many of our clients actually will come to us and ask us to basically provide some support and sometimes guidance to them in terms of how to pair antibody with the payload or a payload plus linker. So that's at discovery stage. The CMC stage is even more so. Gary and Jean both mentioned the complexity of radiopharmaceutical and the CGTs. And ADC is also somewhat similar. We said that developing an ADC, it's almost like developing 3 drugs. Right? You have basically 3 different drug substances in the antibody intermediate, in the linker payload as a small molecule, and then the conjugates. Right? So you have 3 drug substances, and then the final drug product. So in a way, you can almost say that developing an ADC is like 3 times of the work of a typical small molecule or large molecule. And there's again a lot of the cross-discipline challenges when you have biology and chemistry putting together in a single molecule. So we help our clients basically to integrate all of those different expertise together into one single platform so that you can move the asset to the clinics as fast as possible. Traditionally, many companies, if they work on ADC, they will need to basically contract multiple service providers because each service provider has different expertise, on the large molecule or on the small molecule or on the conjugates. So they might contract up to 3, or even more, service companies to do this different part of the work. But it takes a very, very long time. What we do is by integrating all of the capabilities into one single platform, we basically do all of those in parallel. So for a typical CMC, ADC CMC work, basically we will have what we call the 3 sub-teams. Sub-teams focusing on mAb intermediate, focusing on linker payload, and then another one focusing on the conjugation. So the 3 sub-teams move in parallel, and that's the only way to able to move the asset to the clinics at the fastest possible speed. For example, we call that from DNA to IND in less than 15 months. If you do it in 3 separate parts, it could take easily 2 years, 3 years, or even longer. But by combining all the expertise together under one single platform, you can greatly shorten the time. And obviously time, is the most important asset for drug development because your first in class otherwise would become, maybe second or third in class if you don't move into the clinic fast. So that's extremely important in terms of, time to IND. And there, the integration by having a lot of expertise there can greatly, help clients to achieve that critical milestone. And then later on, when it gets to the late stage, there, I think it's even more complex. Obviously not just for ADC, for any, products, when it gets into late stage, when it gets into the BLA stage, there's a lot of very strict requirements, regulatory, in terms of the CMCs, because there, typically it takes about 3 years or so. You need to kick off the BLA preparation about 3 years before you anticipate the approval. The work itself leading up to the BLA submission can easily take about 2 years or more, because there the most important event is your we call that PPQ, basically process performance qualification, or the traditional term is called process validation. So you have to demonstrate at the GMP scale that your process and your product is robust and of high quality. But before the PPQ, you actually need to do process characterization to demonstrate you have a thorough understanding of your process, and then that it can withstand some perturbations in terms of the parameters. But PPQ itself is not just about the process. You also need to basically qualify your facility. You need to qualify your equipment, your manufacturing equipment. You need to validate every one of your analytical methods, and you need to qualify your QC laboratories. So those all need to be put together and ready before you run the PPQ. Right? And PPQ itself, it's almost like the college entrance exam, like the Gaokao. Right? You do a lot of the legwork up front, and then you run that 3 times or more to make sure that it's consistent. But BLA, it's again much more than that, right? Because after BLA, before the approval, there's always this pre-approval inspection, or what they call sometimes a pre-license inspection. It's like the the 面试 basically, right? So you do all of the work, you have actually got high grades, you submit everything, but then FDA or MNPA wanted to come to your shop to see you actually do everything one more time, and they ask a lot of questions. So the PAI or PLI itself is actually hugely challenging. I still remember when I ran the first WuXi Biologics, BLA and tried to get ready for the inspection, it took the whole company about 1 year to prepare, because that was the first, FDA submission from China in terms of the biologics. It took a lot of effort to make sure you have the whole system ready for the inspection. So this has a lot to do with basically pharmaceuticals are highly complex, especially biologics are even more complex. And what we typically see or what you test in terms of quality is just a tip of the iceberg. So in a GMP, in the compliance world, we have a very interesting saying that you are basically guilty until proven otherwise. So this is very different than, for example, drug discovery or any other type of scientific work. What that means is that you have to demonstrate to FDA, you have to keep records of everything. You have to have data that's traceable to say that you actually, do what you meant to do. And otherwise, if you miss any of those components, FDA might not believe you, might not grant you the approval. So that, guilty until proven otherwise kind of mindset is actually very, very important and make it also very challenging for quality, for compliance. And many people probably know about the term called data integrity. So what that means is that every piece of data you generate during GMP manufacturing needs to be traceable. And FDA can, at any time, come to your laboratory, come to your piece of equipment that does the testing to ask you to show, where the original data is. And you have to also demonstrate that that data cannot be easily modified, right? It has to be authentic. So there are a huge amount of those kind of very detailed requirements and regulations that governs, pharmaceutical GMP operation. And those, I think, are not something that you can learn from the book. And many people in the biotech industry, in a lifetime, probably in their whole work lifetime, they probably go through one or twice of those kind of experience. Many CMC people actually also don't get the chance to go through the whole process. And that's why, CDMOs that has a lot of track record are extremely valuable as a partner to help companies advance their, product to the market. So it's a little bit long, kind of sharing, but I hope that it gives a glimpse of, the challenges and also the importance of CMC during the whole drug development, from discovery stage to IND enabling study, and then to late stage BLA submission, as well as to inspection and GMP compliance quality requirements.
That's a very comprehensive review, and also tell people, we'd better leave this very challenging, make or break function to the professionals like WuXi. With that being said, I think, like Jimmy, you correctly identify a lot of audience actually running their startup, and most of them come from small biotech. And obviously, we all know, especially for this three modality, is very challenging for small biotech to really carry their asset all the way to the approval, even commercialize or even manufacture it themselves. So vast majority of those assets are going to be handed over to big pharma, which, obviously going to involve a lot of global deal making. So, that's going to be our second topic today. And the first question is obviously, probably Jimmy, you can continue. As I heard, you involve a lot of the ADC out-licensing deal as a CMC support, CRDMO support, especially for those based in Asia. So can you share with us what's a typical due diligence process when those small biotech, when they get involved with the MNC from the aspect of CMC? And do you have any kind of a case study? Of course, we don't need to know the client's name, but if you can share with us, do or not to do, that would be great. Thank you.
Right. Yeah, certainly. I mean, for any kind of those kind of deals, either in terms of licensing or complete merger acquisition, due diligence is an important part of the process. And obviously, we're not trying to say that other part of the due diligence is not important. For example, the clinical data obviously is very, very critical, right? But CMC plays a hugely important role in any of those kind of due diligence. Especially when big pharma takes over an asset, a lot of times the assets are already in the clinics, right? So there, the pharma or big biotechs, when they do the due diligence, they wanted to make sure that wherever the drug is manufactured, they are very comfortable with the quality system, because they might need to continue to have the drug supply from the current producer, so that they don't interrupt the clinical programs. But big biotechs and pharma companies tend to have very stringent requirements on CMC. So they need to make sure that what they see basically meets what they expect from their own programs, right? For example, their own in-house programs, how they run the CMCs, what are the requirements that they place on CMCs, the quality system, whether it matches what they expect, to pass the FDA requirements or other regulatory requirements. So those are obviously very critical elements that any pharma due diligence people will pay attention to. And typically, when you come to quality, they have the complete sort of authority to veto. Right? It doesn't matter how good the clinical data might be. Doesn't matter how good the asset might be. But if they are not comfortable with the CMC, if they are not comfortable with the quality or the compliance, then they don't have the confidence that they can move the asset in their own hands to continue the clinical trial, for example. Right? If they don't feel that they are comfortable with the CMC, they might have to basically move to another place, for example, even if they really like the asset. And that obviously will potentially significantly impact the clinical timelines. So I think in our case, actually, because of the number of pharma, big biotech clients that we already provide service with, many of the times they are already familiar with us in terms of the CMC expectations, in terms of the quality systems. So multiple of the times we actually were not involved in the due diligence, meaning that the pharma or big biotechs, because they are so familiar, so they know us so well, they simply skip the due diligence on the CMC part. And so in those cases, we are actually the ones that were told, or we found out from the news that, one of the assets that our client work on is actually acquired or being licensed out, because the pharma basically just skipped that due diligence. So that obviously helps a lot, when a client has that kind of strong support in terms of the CMC part. It makes it so much easier for the client to basically accelerate the due diligence and hopefully, seal the deal as quickly as possible.
That's a very good point. So I actually right now want to bring Gary into this discussion, since you just involved one major M&A, probably so far in the past 2 years, the largest radiopharmaceutical M&A. What's your first-hand experience when you do M&A? What the big pharma, in this case BMS, was really looking at? Which stone the particular turn multiple times?
Yeah, without getting the details, yes, I think CMC definitely is one of the key aspects of the due diligence during the M&A discussions. If you just look at all the recent acquisitions beyond RISBio, right? So Point Pharma by Eli Lilly, I think, and then Fusion by AstraZeneca, and you mentioned last week, Mariana by Novartis. These are not a typical asset acquisitions. If you think about, most cases in the pharmaceutical field, right? So large pharma buys a small one, most likely they buy the product. They don't need the infrastructure. They don't need the people. So those are the typical asset purchase. But in the case of radiopharmaceutical, it's actually more about the platform, the radio isotope supply, and then the personnel, the knowledge, the expertise. So definitely the CMC plays a role, and way beyond that is the whole supply chain, the whole basic drug from discovery to development, because every step of the process, there's some uniqueness for radiopharmaceutical. A lot of times, maybe with the exception of Novartis, other large pharmas have limited experience, so they want to have their company that they acquire to actually carry the work forward. So, and there's a lot of sort of discussion about getting access to radioisotope, especially for certain isotopes. There's very limited supply. By buying into the small biotech, they sort of also buy into some of the existing deals, so they have easier access to some of the isotope. These are all the considerations, I think, at this moment. And going forward, maybe once the infrastructure is well built, you're going to see some of the more typical asset purchases. But right now, it's mostly about infrastructure and platform.
Yeah, that's a very good point. So this reminds me of one phrase I keep hearing in the past couple of years regarding this three modality. They always say when big pharma work with the biotech about ADC, radiopharmaceutical, CGT, the process is a product. So this time I will ask Jean, can you please expand on this phrase, the process is a product? Probably you can use CGT as an example. I know you haven't announced any deal yet. Probably you have interacted with the big pharma. What's your sense of that?
Yeah, sure, Liam. So yeah, in cell therapy field, we do see a lot. Process defines the product. Although I think for innovation, drugs like iPSC cell therapy, we're still in the process of understanding the CQA and CPP, and even after 1 or 2 years, when we look back and understanding previously we have with the products, sometimes we found, now we can much more understanding towards their quality and safety and even potency. So it's a long way. So I have to say we are always talking with the big farmers because we know we need partners, especially for oversee development and clinical and commercialization. But we found since it's too new in the field, so they also are lack of the concepts about those products, right? And so basically most of farmers, they don't have their in-house capability of carrying those, iPSC cell therapy products. So when we talk to them, I can't say who. So they were saying, eventually we won't build up the facility or capacity for holding the manufacturing. I say, yeah, we don't expect to, we were thinking about, working with CDMOs in US and Europe. And when we do the tech transfer, preparing for early stage clinical trials, and then, for cell therapies, it's a long way to develop the final process. Especially at the beginning, you have to think about the cost and readiness of your suppliers and automated closed system, whether it's available for your specific product or process, right? So we have been 3 years working with different suppliers and third parties, and now we can say we try to minimize the tech transfer hurdle by developing closed system automated processes, so later on it can be easier. For now it's too complicated process that we can do stably at our facility, but if you aim to transfer to CDMO partners, or eventually the farmers will be in charge of the financing and clinical trials, it can be a disaster. Yeah, so we understand now even for formulation tech transfer, it takes 8 months, not including, you know, all the analytical method transfers. It's only about limited releasing testing methods that can be transferred for that part. Comparing to the whole manufacturing process and complexities, it's nothing. It's only about 5% of the work. It takes 8 months. Yeah, so we're thinking, so that's you know, general idea. The pharmaceutical companies, they have to make sure you reach to your certain stage of clinical trials first. It's like proof of concept of your product, right? And also the manufacturers, they understand, especially for Bayer and others, they have acquired iPSC cell therapy companies. So they know if they step in too early, they're going to develop the whole manufacturing process, quality, and everything together. And they don't have the expertise and that time in their hand as well. So that's what the biotechs, you know, we aim to push it forward and then work with our partners. And we have the knowledge about the product understandings of current methods that we can utilize to understand the product, you know, quality and properties. So eventually at a certain stage, when they are confident to take over and the whole system is more mature, I think that's where, you know, work together with the CDMO altogether, we can make this drug, you know, towards pushing towards the commercialization. Yeah, that's my idea. Yeah. Mm-hmm.
Any comments from Jim and Gary from your you know, expertise in ADC and radiopharmaceutical respectively in terms of the phrase, the process, the product? What's your view on that?
Yeah,
No,
I think
it's, go ahead.
okay, sure, Gary. Yeah, it's a phrase that you know, all of the biologics field actually hear very often, and that's also the reason why in the small molecule field you have the generics that basically are completely interchangeable when the innovator basically expire patents. But for biologics, you only have biosimilars because it's almost impossible to get the identical, and that has a lot to do with processes, obviously. Every single process or even transfer of a process from one place to another place could lead to some subtle differences in terms of the product quality, right? And so that's why you always hear this phrase, process, you know, is the product. And that's the inherent challenge of biologics, and obviously also applies to ADC because there you have the, you know, mAb intermediate. So that makes it even more important in terms of CMC to make sure that you have a thorough understanding of your process, that you demonstrate your process is robust, and also when you scale up or when you transfer from, you know, one facility to another facility, that you have all of those, you know, quality attributes. You do those compatibility assessments to demonstrate that, you know, the products are actually equivalent or what they call comparable. I think it's the fun part of you know, CMC work. It's also why you know, CMC experts you know, become more and more valuable you know, as they experience more and more of those processes.
Yeah, just want to say briefly, the process also the barrier for entry. Actually, you know, for ADCs and even more so for radio, because if you think about the current product, a lot of the you know, the initial binders were developed a long time ago. You know, the patents already expired. So literally everybody can take the same binder. There's no strong protection of the IP. You can, you know, in theory, you know, link to your linker and payload, and you have a drug. But it's not that simple, right? So the barrier is really, you know, yes, you can have that, but how do you get the supply chain going? How do you build the facilities? Considering the radioisotope, you need a lot of more sort of regulatory process to go through. And then also I mentioned earlier, right? So the almost on-demand requirement of the drug. So a lot of those barriers that's actually very difficult to overcome. That's why you see a lot of, you know, talk about radiopharmaceutical, but reality is it's very expensive and difficult to get into the field.
Yeah. I just wanted to add, one more quick comment on this, Leon. ADC at this moment still does not have a biosimilar yet. So that also, says a lot about the complexity of the ADC CMC. Is that if you ask FDA to try to define what the ADC biosimilar will look like, it's actually a huge challenging task.
All right, fantastic. We already saw a lot of questions, so we're probably going to skip the last topic, which is a critical, unique feature of each modality. But please keep that in mind when you address the questions. You can fold those unique feature of the modality you are expert on into your response to those questions. So the first one, this one is from David Dye. I believe he's from UBS, which is an investment bank sales side analyst. Question for panelists, given that lead only have 10 hours of a half-life and will need to be delivered to the patient within 5 to 8 hours, how practical is it to manufacture lead-based radiopharmaceutical in the morning and deliver to the patient on the same day? What will be some key CMC improvement to make lead-based radiopharmaceuticals more practical?
Yes, that's for me. Yes, that's how it's done. You manufacture and you have about a day, work day to deliver. You just have to plan ahead. Also, there's a concept of disseminated manufacture. You cannot have a central manufacturer. You have to, in the case of US, you might need maybe 10 or more manufacturer pharmacies, for example, to be able to manufacture, and then you deliver, to the hospitals within, maybe 2 or 3 hour driving distance. That's a reality for lead-based therapy. But for lutetium, actinium, they have much longer. You probably can get away with a central manufacturer and ship globally.
Fantastic. So, by the way, sorry for the mispronunciation. I didn't realize he was meant lead instead of lead. Anyway, so that tell you everything about my chemistry background. Another question from Yang Yu. Could you please discuss how WuXi approached the global supply chain challenge in the ADC manufacturing process, including map, linker, and the conjugation stages? I think it's mainly about a global distribution supply chain.
Right. I can take on that. Yeah, ADC, because of the special nature of those highly potent, highly toxic payloads, the supply chain and the global capacity is actually very constrained, very limited, because you cannot tap into the traditional biologics facility to manufacture ADC for both the conjugate drug substance as well as the drug product. So basically, we have to continue to build. You need to build a lot of capacities. So we have, in terms of XDC, we have built that in Wuxi City. We are also building in Singapore, and we are also actively looking to, further expand into Europe and US. So that's one of the challenge for the industry is actually, the capacity is very much limited. Leo, I think it looks like you're muted.
Oh, how about now? Sorry about that. So I should have moved to this slide. So once get into Q&A, I know a lot of people dial in from other places of the world did not register this. If you want to get a replay, please of the webinar today, please scan the QR code and offer your email address. We're going to send the replay to you after this, before we post it on the link. This kind of incentivize you to become the insider of our webinar system. With that being said, the same thing. If you have any question, please put into the Q&A box of the webinar. Okay, so there's a few more, and let me just there's another question from Mike Orsini. I believe he's probably from Daiichi. For CAR T therapies, are there any particular CMC advantage or unique consideration depending on cell type? For example, NK versus T cell or allogeneic versus autologous therapies. I'm not sure people can answer because probably you are the expert. So you can give a try. If you don't, I can try to cover.
Yeah, maybe I'll give a shot. Yeah, so actually, I think from CMC aspect, it's more relevant of whether you're developing an allogeneic or autologous, immunology cell therapies, because it can be quite different from logistic and how you handle the process and the virus and the cost, right? But from the CAR T or NK, whether you're going to link CAR to the NK, that's also one thing. And how hard you can reach efficiency of expanding those cells, and whether T cells can be used for allogeneic cell therapy. That's also one consideration as well. Yeah, so we have been developing CAR NK cells from iPSC for a while, so we kind of know the area a little bit and how strategies or debates over those. So it depends on the cell type and the indication, and what's your technique and what's your process, and your original source of cells also matters. Yeah, so it's quite a big question. It takes maybe at least 20 minutes to fully address that, I guess. Yeah. Okay.
I guess this audience could send an email to us if we can forward to you if you're willing to give her a little bit more color. Now let's get to the last question. This is from Denise Chen regarding radiopharmaceuticals. Given the lead for speed of delivery, ideally local delivery, does a high population density in China, urban centers, lower the logistic barrier and improve the commercial opportunity? For example, should a radiopharmaceutical developer be thinking about China as a particular attractive market? Or in this logistic, actually it doesn't matter. I think this is for Gary, and others please chime in as well.
I've seen. Yeah, it's going to be a long answer. So I'm really apologizing because I need to jump to another meeting. So I can leave my personal email to the webinar. Maybe you can have a separate discussion.
I think Gary is when we had to answer on the call. That's what we were doing. Okay.
I think someone is talking. Can you please mute yourself? But anyway, this ends the webinar. Thank you very much. Today I really enjoyed the whole thing. I learned so much. With that being said, thank you all the audience around the world dial in today's discussion. And also a little bit, pre-announcement. We do have an ASCO recap. I believe in June 6th, right after ASCO. And that's actually quite a historic webinar. Why is that? That's going to be our 12th webinar in this series. That's kind of full year. That's kind of an anniversary for us. So as you could imagine, we have already have quite a few pretty influential speakers lined up. And this year's ASCO going to be very exciting as well. And I personally going to be there, and hopefully it can be the sound of our audience and some of the members from Insensible Trust community, as well as other collaborating organizations over there. With that being said, thank you all for your time, and thank you all the speakers again for the fantastic sharing. I will see you next time, and take care. Enjoy your day.
Thank you,
Thank you.
Leon.
Bye.
Thank you. Great pleasure.
Bye.